after. So thanks everyone for coming, and big thank you to you, Andre, for sharing your work with us today.
Yeah.
So give it to you.
Yeah. So I was asked to talk about something I've been working on, and as I just said, I used to do research, and then I reviewed research, and now I just pay for research. But occasionally I do some work. And this was a little project that I got involved with during my time down in Australia, actually last year, not this, well, 2023. It's a little problem that I hadn't thought of before, and it's one that crops up quite a lot, which I'll outline in a moment. But basically, it's what do you do if you're doing a stock assessment and you don't know what species your catches are? And I'll talk about why that can be a problem. The other cool thing about this is it's probably the last project I will do with the third person listed at the bottom of the page. The co-authors, this is a talk where you emphasize your co-authors. So like my previous boss, or one of my previous bosses, his claim to fame was he wrote a paper with Robert May, right? You know that name, the Robert May. So the paper is et al. May, right? Who the hell cares about anybody else? Anyway. The cool thing was this was working with Bill Venables, who you might have heard of one of his packages. I believe it's called Mass. Ever come across that package before? And the other cool thing about working with Bill is if the code doesn't work, he just changes R. Anyway, this is all about a fishery I've been, as I said to few minutes ago, I've been working on since the mid-1990s. This is a trawl fishery for prawns in northern Australia. You can see, where's my mouse gone? Do I have a mouse? I do have a mouse. So we're talking about this region of Australia. It's, look at that. It's a very remote part of the country. The only city of any note up there is Darwin. If you've ever been to Darwin, you would barely call it a city, but it is de facto the largest city in the Northern Territory of Australia. It's a very big area, and it is Australia's most valuable Commonwealth managed fishery. So Australia, most of our fisheries that are worth more than 12 cents 95 are invertebrates, and they are managed by the state. The exception is this northern prawn fishery. I'm going to refer to the Gulf of Carpentaria quite a lot here. So that's the bight that comes out of the top of Northern Australia. Most of the fishery is in this area. And then there is some catches of various species in what's called the top end, which is the piece over here. And I really won't be talking too much about catches in what's called Joseph Bonaparte Gulf. Does anyone remember who Joseph Bonaparte was? Napoleon's brother, who was the king of Spain, not necessarily voted by the Spanish, was thrown out, but ended up dying in North America. Anyway, so this most valuable fishery in terms of product, it produces a lot of premium prawns. So it's not just, it's a fairly large fishery in terms of volume. It's about 3,000 to 4,000 tons. But it is premium product. No Australian gets to eat prawns from this fishery. That gets flown to China and Hong Kong. There are going to be three main species groups. I'm going to talk about two of them. Tiger and endeavour prawns are going to be the focus of the talk there. The long-lived prawns, so they get to be a second year. Banana prawns I'm not going to talk very much about at all. And I'm also not going to talk about the challenges of managing a fishery where probably about 95% of the catch is bycatch. And this is not, Dana's looking shocked.
That's so much, I didn't know.
Oh, prawn fisheries, that's not bad at all. It's shocking how much, and it's just small, completely worthless stuff that gets thrown over the side. But thankfully, it's very productive. So the biggest bycatch species we actually worry about are sea snakes. Which personally I have no conservation priorities for. Anyway, So what are we trying to do? So, this is a MSc certified fishery, so we have to do stock assessments and provide management advice in the sense of what is biomass relative to BMSY and various things like that. So we've developed some stock assessment methods. They tend to be based on size structured population dynamics model rather than age, because you can't age prawns. The idea is that we have to do stock assessments by species. That makes a lot of sense because that's what we're reporting on. The problem is that the catch is recorded in logbooks by species group. So instead of splitting, and I'll talk more about the species themselves in a moment, but instead of splitting the tiger prawns into their constituent species, they just record it in logbooks as tiger prawns, right? This is the complication. So how do we split those catches into time series that we can then put into our stock assessments? The science behind the monitoring, like we got surveys and stuff like that, that's all done by species, but the actual fishery is all in these species groups. And as I'll show you in a moment, if this was the West Coast ground fishery, which Alan remembers well, this is not an uncommon problem. We've got unidentified rockfish, which need to be split species. So you go to some port and you say, well, we've got 20% is X species, Y species, and we just multiply out, right? And then we hold our breath that we got it right. The difficulty with these guys is that the species composition is a function of the ecology of the animals. So as I'll show in a moment, the species split is going to differ between locations within that Gulf area. It's going to differ between days of the year in the same spot. And part of that is the behavior of prawn. So one of the species that I'm going to focus on tends to dive into the substrate when it gets warm. And so basically catchability changes even within a location over time. Across the year, it changes. And then potentially it's changing over time as species change their distribution. So what should we do? Well, we know the answer to this. It's very easy. Instead of doing all this fancy math, we should take each catch record and tell the fishermen to disaggregate their catches. Well, they're not going to do that. Our data are recorded by 6 by 6 minute grids, and you'll see that there are literally thousands of 6 by 6 minute grids in that Gulf of Carpentaria. And so we need to sort of come up with a model, a prediction model, to be able to essentially say, I caught a certain group of prawns in a particular area on the 7th of March, 1960. Not 1960, we didn't have a fishery yet. 1975, what is the split of that catch to species? So we're working at the level of a grid cell on a particular day. And so the question is, how do we do that?
Andre, and
yeah,
you can easily visually tell these species apart?
Scientific. So only observers really do the, and I'll show you the data that we're using, but yes, that's one of the problems. And also obviously they want the product to be, they don't want biologists fiddling around. Mm-hmm. So this is some plots of, these are actually model predictions, but each of the cells that you can't see on the right-hand side is a grid cell. So there are lots and lots of grid cells. And I'm just showing some model output for 4 days. There are 365 days in a year, it turns out. So there are a lot of predictions that we have to make in order to get this to work. So the questions I was confronted with is, I'll talk about it in a moment. Previous work has essentially assumed what's called a static model. So in other words, if you go to a particular cell on a particular day, the proportion of whatever species you're dealing with is the same over time, right? And I'll show you why that's not a completely crazy thing to do. The question is that true? The second is, we've got a catch time series that we've been using in our stock assessments. The good thing about models is you can create a whole new model that changes absolutely nothing. Does it actually change anything we care about? And And then this fishery is, I believe, the only fishery on planet Earth that is modeled to maximize profit, as opposed to most fisheries the target is the MSY or something like that. This fishery has been managed for the past close to 20 years to maximize profit, and that does give an idea of one of the differences between the Australian, dare I say, New Zealand and US systems, which is it's called user pays. So the government doesn't pay for the research, the fishermen pay for the research. And so making money is the aim here. There are no subsidies in these fisheries, right? If you can't make money in the fishery goes away. It's not quite as crazy as New Zealand, but it's doing its best. So those are my three big questions. So how can we solve this problem? This problem's been recognized since the fishery started in 1969. Literally, we know every single trawl in the fishery that has ever occurred. The fishery actually started with a research survey that found prawns. Literally, no one knew these animals existed, and they did just straightforward species survey and found prawns and developed the fishery in 1960. The first catches in 1969, 1970. So the idea was a guy called Ian Summers, who was the original biologist who essentially started the fishery. He's now a multi-millionaire because he realized the error of his ways and stopped being a fishery scientist and became a property speculator. And it turns out that if you're a grad student, buy a house in Seattle and sell it 20 years later, you're doing a lot better than your job at NOAA. Sorry. Yeah. I mean, that's life. So the idea, what he did was he had some survey data and he did spatial interpolation. So basically where he had data from research surveys, he would use that split for that particular cell and then extrapolate it, just sort of using a nearest neighbor type approach, right? So if I've got data for this cell, I'll go to that next one and use the same split and different weeks, and it's completely unreplicable, but everyone believed Ian. The problem is with that, of course, is that there is no uncertainty in your species split. So if you only go to one area and you get one sample, you're going to treat that as if it's known perfectly, right? So the advance on that was work done by Kathy Dickmont and Bill Venables in the early 2000s, where they fitted a GLIM. Admittedly, it was a GLIM. So for those who don't know, GLIMs in the 2000s didn't have the S function that you do now. And so Bill literally wrote that. This was a GLIM with some splines. It was a very innovative analysis, but really didn't address the key questions that we were interested in today, which is those three things that I was after. Have things changed fundamentally? So in order to address this, so I was hoping Jim Thorson would be here because I could see him frothing at the mouth saying that you should use VAST for this problem because VAST is the hammer, right? And so if you know the analogy, if you've got a hammer, everything is a nail. So you've got VAST. Anyway, so that was my original idea, was to say, well, we're going to do a geostatistical analysis. Well, I got an F grade for that from Bill Venables for two reasons. And the first of the reasons, and I'll go through a little bit more detail in a moment, is the plot on the left, which is the data that we've got to do species split work. So we've got data going back to essentially 1975. And what this plot does is shows you the temporal resolution of the data that we've got. So we've got most of these sort of going back to Alan's point, most of these are research surveys where we went to particular places in the Gulf or the top end of the Joseph Bonaparte Gulf and collected some data, the size of the dots, the proportional sample size. Most recently, you can see those green dots there. Those are actually research surveys that we've got data from. But you can see that the surveys are quite short in general because they were specific funded research programs. The work, this project entailed not just modeling, but also you can see some green points down at the bottom, AFMA Observer and Species Project 2. There was dedicated observers placed on fishing vessels to collect data, to get a bit more synoptic data collection across the Gulf of Carpentaria. But it's a very unbalanced data set, right? And I'll show you in a moment, it only gets worse. I'm going to refer to some areas within the Gulf or within the fishery. So there are 7 areas. Most of them are going to be in the Gulf of Carpentaria, GOC on the right-hand side. There's 2 in the north. There's Arnhem, which is an island, and Melville, which is also an island, and then the GB, which is Gulf of Joseph Bonaparte Gulf. So when I do refer to areas, these are going to be the areas. Most of the action is going to be in the Gulf of Carpentaria for these species. Okay, so let's have a look at the data that we've got. So remember I said that there were 12, I didn't say there were 12, but there were 12 surveys that have been conducted. This is the data which we've plotted by number of samples. So red means 10 samples, green means there was a single sample taken. Most of the action is here in the Gulf. You can see essentially no, so this is 1976 to 1979. There's really nothing outside of the Gulf. There are a couple of cases where they've collected data from here in Melville, but you can see most of it is in the Gulf. There's a lot over here in a place called Weipa, and that's actually where a research station is based. So they collected data from the research station. So you know, one of the main problems with this data set is it is so spatially unbalanced, right? So almost all the data is coming from the Gulf and really from this area here in the south west Gulf and the Weipa area. And, you know, there's very spotty data in these other areas.
Is the fishery more uniform?
Most of the fishery is in the Gulf, and some of it is out. For one species is out here in off Melville and that area. Yeah.
Question. Looks like there are fewer samples 2018 onwards, but it looked like in the previous graph that you showed that there was a pretty high sampling intensity. Like with
Well, remember I'm aggregating over decades.
Oh, okay.
So that's, yeah, so
Okay.
there's much more data in the 2018s onwards where we've got dedicated sampling. And this gets to Alan's point, you know, where there's no data, that really means there's no fishing. Yeah, and that is going to come into play. We don't really care about predictions where there's actually no fishing. Not a concept we tend to see in other areas, or huge fractions of the fishery just aren't fishing. Okay, so what is the problem? So this is some data again. This is looking at tiger prawns. The problem is actually not as difficult as it might seem. There are many areas where we're absolutely certain what the species is, right? So it is definitely one of the 2 species. And for example, I don't know if you can see, there's an island here. This area essentially is one of the 2 tiger prawn species. It always is, always has been. So we don't need to really focus on those areas. We're focusing on the areas where there is an actual split that comes into play. And so that makes it simpler and also changes the focus for some of the work. And as I say, so what we're looking at is these split areas. So what did we do? We're going to fit a model to those data with the idea of predicting. So this is where VAST starts to really become challenging. So most of us have run VAST and STTMV. Most of you have produced a plot by year, right? All you have to do here is plot a plot for every day from 1970. That's a lot of output, right? So what were our data sources that we're going to put in the models? I'll tell you about the models in a moment. So we had basic lat long as sort of initial covariates, but we had a number of, we involved sort of the prawn ecologists in terms of what we know from experimental work. Good thing about prawns is you can actually do experiments on them. They only live for 2 years. The types of things that we knew affect the species composition is the substrate. And in particular, mud, the proportion of mud in the substrate. We also looked at a couple of other things, the grain size of the substrate and benthic stress, whatever that is. And then a number of other factors which I'll go into. So this is the distribution of mud across the area. You can see this area up here being much more muddy than down here. This is sand. So the other covariates that we looked at are the first one, let's start with that one. This is distance from the coast. So we work out the minimum distance from any location to the coast, which is why you get this psychedelic. Yeah, I was actually going to use psychedelic colors in the paper, but eventually thought that probably not a good idea. Then we got depth. So we were hearing from Curry last night about his species. You can't quite dive for these guys, but the Gulf is actually really quite shallow. And in fact, one of the more interesting sort of socio-dynamic things is Aboriginal people remember settlements in the Gulf of Carpentaria. Admittedly, we're talking tens of thousands of years ago, but within people's memory, because this area really is very shallow, and that's how people got to Australia. Anyway, so that's depth. And you can, if you remember the plots you've seen before, almost all of the data are coming from these inshore areas where it's pretty shallow. And then the final one that we had is called R-dist, which is a curve. I can't remember how many points that we've interpolated, but essentially it's a measure of the distance along the coast. So you can think of the covariates in this model as the environmental data, the distance offshore, the distance along shore, and the depth, right? So we got all those covariates that we're going to be looking at. So just a gentle reminder that, you know, the cool thing here was lots of model, not much data. So there's an enormous, this is just a couple of years of examples. You can see in any individual year, there's really not a lot of color in those patterns, right? So we're using data, leveraging data in years, across years and things like that. Okay, so apart from that, what was the next problem? The next problem was that the data, well, I've referred to this as proportion data, right? But if you do a stock assessment, you're not interested in numbers of prawns. You're interested in mass of prawns, right? So the data that we've got are someone takes a grab, pulls up a bunch of prawns, weighs the prawns, and we got a proportion, which is a proportion in weight, not in number, right? So our sample size, our N, if you want to call it that, is the weight of the sample, right? So, you know, before I started this, I said, oh, this is easy. We're going to run a binomial likelihood because it's species 1 or species 2, right? It's not, it's weight. And weight doesn't behave like a binomial. So we ended up, we do define the proportion as, so this is one of the tiger prawns, semi-sulcatus, otherwise known as grooved tiger prawns. So the data in this case is the weight of semi-sulcatus divided by the total weight of all tiger prawns. So it's a proportion, but it's a proportion in weight. And so what we ended up with was to say, okay, well, what is the measure of the quality of the data? Okay, so if this was proportion data with N, the problem is you've got over dispersion, right? So if you've got a whole bunch of species 1, they're not in the sample is not independent, and weight is not N, right? So 6 kilograms. How many N's is that, right? So what we ended up doing is using a quasi-binomial likelihood, which was a first for me. And essentially all it is the variance of your prediction is the mean times 1 minus the mean, normal binomial formula, divided by the weight of the sample, divided by a theta, which is essentially an over-dispersion parameter. So if theta was equal to 1 and t was equal to n, this would just be a binomial, but the theta parameter is accounting for the over-dispersion. And this was my first introduction to ChatGPT. So I thought, okay, I'm sure that the STTMB people have solved this problem. And I went, give me the code for a quasi-binomial for SDMTMB. And it said, SDTMB x tilde y family equals quasi-binomial. And I ran it, and what did it do? Does not exist. So ChatGPT correctly realized that family is the command that you use to define a family, just not in STTMB. So that ended there. So that wasn't so great. So we actually ended up using general additive models for all of this. So everything I'm going to show you are general additive models. They have the advantage that you can use quasi-binomials in a straightforward GAM. It also means that your variances don't mean anything. AIC really doesn't do anything for you. All the model selection stuff in the world sort of dropped out. So that was our next problem. So, because everyone does it, we've got more models than you have. My number of models, lots of tensor splines. So looking at lots of interactions between variables. So we started with a model called model 0, of course, because that's what we all do. That just had a spline. The nice thing about the Gulf, it's close to the equator, so you don't need to worry about Mercator projections that like we do in the Gulf of Alaska and the Bering Sea. You can almost walk to the equator from the fishery. So we started with a straightforward spline, lat long, and then we brought in tensor splines to account for potential interactions. We didn't do every single potential. We certainly didn't get in triple interactions, but we did set up two sets of models. The first set of models, which I'm going to call initial models, are models that have do not allow for changes over time in the split of species within a cell in a particular day. So it's what we call a static model. So what you're trying to do is you're trying to find an overall split for 1 year that you're going to apply to all years. And part of that is you just don't have a lot of data, right? And then the non-stationary models, the main change to the model was adding in a spline in terms of days since 1970. So the 1st of January 1970 was 0, and every day thereafter was one more than that. So think about it as a spline adjusting the expectation of the linear predictor in a GAM. And then we asked the question. So this is a model that allows for changes over time in species split proportions. And then we said, well, what happens if there's been changes in how species interact with the environment? So these other tensor splines say essentially, let's take this one, that the interaction between the depth and day. So basically, over time, the effect of depth is changing with respect to species, right? So can you see where I'm going to? So these are essentially probably climate-driven things that we were looking at. So yes, these are quite complicated models. They take a while to run. They don't always work, but that's life. So this was where Bill Venables sort of came into play. Bill is truly amazing. We all learn how to do statistics. He's an artist. Statistics is here to solve a problem. And if the math isn't quite what you want, it's good enough. It's got to be good enough to solve your problem. And only someone who's a professional statistician can say, just because the residuals don't look that good, they're good enough. I've seen lots of residuals in my career. This will be fine. So what we were aiming for was to come up with a model that was good enough for its purpose, right? The residual patterns aren't going to be perfect, but our aim here is to do a stock assessment. We're not going to reject the model because we're going to stop doing stock assessment, right? So what did we do? So the things that we looked for were, how much of the deviance did we actually explain? Noting that the absolute level of deviance has no meaning because it's quasi-binomial. That scale parameter theta, in other words, the smaller the value of theta, the closer you are to a true binomial process. We looked at some residual distributions. We had to fiddle around because it turns out you can't calculate those. And then we used cross-validation, which at the end of the day was probably the most powerful way to evaluate a model where the absolute value of likelihood really didn't mean anything. So what is it? So what is a residual? Okay. We got thousands and thousands of data points. So who went to the think tank last week? Right, Pearson residuals, right? Okay, forget the fact that, well, actually they do apply here because this is a fixed effects model. So in theory, all you have to do is you take your observation, you subtract your prediction, and you divide by your variance, right? That's your square root of variance. Most of my data is zeros and ones. Okay. I could, if I thought about it, have used residuals, but I didn't. So what we did instead was we essentially created pseudo residuals, where what we did was we said, take, you've got to think about the data set. You've got, for every day from 1970, you've got a sample, right, on a particular day. What we did was we said, let's just add up the data until you reach, I think it was 6 kilos of data. So take your data in time and just add it up so you've got 6 kilos of data. And do that through the next 10,000 data points. And what we ended up doing, this W thing here, essentially we defined the observation not as an observation at a particular point, but an observation over a sufficient amount of data that you would get a proportion that actually has meaning. So only someone who's the high quality statistician can get away with that with reviewers. The rest of us would get rejected and told to do it properly, but you're Bill, you can do this the way we did. So we essentially created a new form of Pearson residual that worked. And why did it work? The question we were answering was not, is our model perfect? Because it's clearly not. What we were interested in is there a trend in residuals with time? Okay, so in other words, if you, for your problem with residuals, you get thousands of numbers. You're not going to be able to see anything. So I'll show you a plot of this, sorry, it was 2.5 kilos. These residuals, which are going to allow us to ask the question, is there evidence in the model for changes over time in species split proportions? This issue of that days variable.
You weren't interested in issues over space?
Oh yeah, no, we produced thousands of them. But the most of it was best visual fit, to be honest. It looked good enough.
Yeah.
And if you increase the font size, it looks so good. It's like the old trick with MCMC diagnostics. If it doesn't converge, just increase CEX equals 2, and it gets blacker and blacker and blacker, Okay, so the cross-validation, we actually did quite a lot of cross-validation. One, we just did the sort of standard cross-validation where you leave 10% of your data set out. That sucks really, because these data aren't independent. So we did it. And then we summarized the performance by the mean absolute difference between the predicted proportion and the observed proportion for the data set. So that we did that because that's what everyone does. But what was more useful was to say, if we remove an entire data set, right? So remember those data sets going through time? So basically, let's remove all the data from the 1970s and see how well we predict the 1970s. So we're trying to deal to some extent with the independence here. So we focused here on the most recent data sets, which were the ones that we thought we were most interested in, but we'll see in a moment we were wrong. And then we also, the great thing about the survey, so the fishermen use the survey as a planning tool. So it's really expensive to take a boat into the Gulf of Carpentaria, because most of these boats are either coming from Perth or from Brisbane, right? So you don't want to be Alan taking your boat all the way from Brisbane to the Gulf of Carpentaria, only to discover there's no prawns. So we do a survey before the fishery starts. And so we get lots of data from the survey. Not very helpful for species split because that's before the fishery stops. So we also did some sensitivities, leaving out data before the fishery actually would have commenced. So basically focusing the data when there's actually something going. So what did we find? Our explained deviance was actually, I'm now going to show results separately for tiger prawns, which is actually the biggest component of the fishery, and for endeavor prawns. Tiger prawns are going to be, there are two species. There is what's called grooved tiger prawns and brown tiger prawns. Don't ask me why. The good news with endeavor prawns, they'd run out of smart ideas. So there's red ones and blue ones. And they're not red and they're not blue.
Never.
They're never on. Hey, normally what we would have done is named them after some species in the UK. That's what we usually do. Like New Zealand's got whitings that don't have any relationship to whitings, but
Dover song.
yeah. Anyway, so what happened, what we found for our initial analyses was exactly what you would imagine, that the more complicated your model is, the higher the explained deviance and the lower the overdispersion parameter. So that didn't tell us very much. The only time that stopped being the case was once we moved into some of these models that had these very complicated tensor spline interactions with time. And what our interpretation of that is, we're overfitting the data really badly at that point. There just is not enough contrast to estimate how has depth affected species split over time. There's just not enough data in that. You'll see, I'll show you some quite interesting plots that illustrate the evils of overfitting. One of the rules, and this was sort of a message that I sort of took out of this, is rule one of any model fitting exercise. The best model shouldn't be the most complicated model. You should always have a model that's more complicated and gets rejected, because if the most complicated model is the best model, there's probably another model that you haven't found yet. So we wanted to sort of go off the cliff of models. The good news, this is the, don't worry about the percentages, look at the numbers. The cool thing about the cross-validation is generally, as we increased our model complexity, we also reduced the cross-validation score, so the MAD went down. And the exception to that was once we started getting into the non-spatial models. The non-stationary models. So again, this overfitting issue that once you have a model that's too complicated, its performance on cross-validation starts to deteriorate. So we had these sort of two things, that more complicated models fit the data better, but there comes a point where you're overfitting the data and in fact your predictability goes down. And I think there's a lesson there that when we do modeling, we should get to a point where we get models that really are too complicated for our data, rather than just stopping when we get a really complicated model. So these are these weird residual patterns. Remember, they've been ordered over time and then crunched into 2.5 kilos. This is tiger prawns, endeavor prawns, much more data for the tiger prawns. And if you squint, so models 4A, 5, 6, sorry, 4A and 5 are the non-stationary, or the stationary models. So they're assuming that the pattern is the same in every single year. If you squint, you will notice that there is an increasing trend in those in the black line, which is the lowest smoother through this. And then once you move to model 6, which is when you've got a spline in terms of time, you'll notice that essentially what was a trend has now disappeared. So what this is pointing to is supporting the time that for tiger prawns, there is this change in species split proportions over time. For endeavor prawns, the evidence is much weaker. Here are the residual patterns. If anyone can explain why this is endeavors, this is tigers, you get this horrible kurtosis in the residuals. We're still trying to work it out, but we don't care, so that's okay. Paper's being published. What more do you want? Hey, all you have to do is convince two reviewers. What we start next, the next sort of, so we've got a model that works. Does it make any sense? So the first thing we looked at, and you, again, you have to be a professional statistician to get into wiggliness, right? So we actually presented this to the decision makers, and there was a long discussion on whether you put quotes around wiggliness or whether wiggliness literally is a statistical concept. But wiggliness was very important to us. So what we've got here, remember we had those regions in the Gulf, the 7 regions. What I'm showing in this panel is for endeavor prawns, the effect, this is the S, the effect of days over time by area. Now, because this is obviously a binomial model, you can't just interpret the factors themselves because they're all interacting through the linear predictor. So this is a prediction for wherever area 4 is, I think it's Groot. Again, very original, that island that you saw, it's called Groot. How's your Dutch? Because of course this is all, Australia was named by the Dutch explorers. Groot means Groot. How's your German? Groot means big. It's the big island, right? That's literally the name of the island. Of course, we call it Groot as opposed to Groot, which is what's good. But anyway, you'll notice that there's wiggliness, but no trend, right? So the model is estimating a spline, but there's really nothing going on for endeavor prawns. For tiger prawns, you'll notice that their trends, their increases around the 2000s. In terms of, this is the effect, the relative effect of the different species within the tiger prawns. Alan, you're looking very skeptical.
Well, I'm just wondering how much, when you showed your data, it had all these different surveys.
Yeah.
And they were very sequential.
Yeah.
There wasn't much overlap between surveys, and I'm wondering if that has any effect.
So, okay, so that is, I don't cover that here. That is exactly one of our major concerns. That was why we did the sensitivity to leaving out the data. In fact, well, firstly, you don't see it in endeavor prawns. So it's not a completely data thing. But I had this, it smells bad feeling, because this is where the survey starts. And yeah, so that was one of the reasons we said, well, what would happen if we left out like some of the early survey data? And the trend actually becomes more imprecise because you're throwing most of the data out, but it's actually quite robust. But that definitely started the you know, there's something potentially bad here. This is, don't pay too much attention here. This is what happens when you use those models, those overfitted models with interactions. So this here is the same prediction that you're getting there. But now you've got these weird interactions. And some of these patterns just make no biological sense at all. So again, indicating that, you know, we've got to the point where we've overfitted data. We did a lot of sensitivities, going to Alan's point. One of the challenges though is, so everything was fairly robust. But so this is the total number of data points, 30,000 data points. This is the one you're talking about, the monitoring program. That gets rid of 20,000 data points. So you do lose a lot of precision if you throw those data out. But it turned out it was fairly robust. This is actually a full set of those spatial trends when you throw out some of the data. Obviously, this one gets rather imprecise. Okay, so where did we get to? This was a cartoon by one of my co-authors, which was really interesting because that's Aquatic. And this was the presentation to the scientific review group. Does anyone remember where did we use AIC in this talk? Nowhere at all. So no one noticed that they got a picture of Aquatic in a talk that didn't actually use AIC at all. Anyway, that gives you, I won't interpret it. So our eventual recommendation is for tiger prawns, we want to have that model, the biggest model that had a temporal term, but none of the models with interactions. And for endeavor prawns, we adopted a stationary model. So what does that mean? We end up with, I'm not going to go through a lot in terms of predictions. We end up with lots and lots of maps. It is not that hard, not that easy to see differences between, this is actually for a particular year at different times of the year. But if you look here in this area, you'll notice that there is variation across the year in the predictions. A lot of the areas are 100% one species or the other species. And these are just different predictions these were for different years. So what did it all mean? So at the end of the day, I did all this work and I closed my eyes and I took all the data and I said, did it change anything, right? And the answer is yes, it did. And it was very revealing. So what I'm showing you here are plots. I'll show you a better one later on. So I've got these are the two tiger prawns. These are the two endeavor prawns, and this is actually the effort in the fishery. So one of the things in this fishery is that there are two métiers, target fisheries, and we define métiers based on the catch composition. So if you change the catches, not only do you change the catch series, you actually change the effort series. What was interesting was that, and I'll go to more detail in a moment, the series changed everywhere. We thought we would see most of the change here, where we brought in the new data, but in fact, because this is a model, not real data, putting in data in 2000 changes predictions in 1975 because it's a single model, right? So there's a lot of leverage going on in terms of this model. There's also very little data for the 1970s, so there's not much to stop it. So it did change the recent catches. The one that perhaps is most relevant is this black line here. You'll notice the black and they're inverse in the two panels because they have to add up the same thing. You'll notice that there's this increasing trend here, where because of that spline that in days has changed the species composition of the catch quite appreciably. So let's keep thinking about what this means. I'll forget that. Let's go here. So what we wanted to do is to ask the question, where did our catches go, right? So the managers were, put mildly, a little on the surprise side that rerunning a model changed the catches that they've been knowing about for the last 30, 40 years, right? And so what we actually ended up doing was to say, let's take our current model, which is the model that Bill Venables developed in 2006, and essentially change the model structure to attempt to work out what feature of the model and the data is actually leading to these changes in catches. So model 4A was used as the reference for this. And what we found, so this is tiger prawns, these are inverse, obviously, these are endeavor prawns, effort. What we found for endeavor prawns, it was data, right? It wasn't the change in the model. The changes in the model, it's hard to see, but most of these plots are on top of each other, or this is 0 means no difference from the base model. These are crazy models. Essentially, the thing that made the difference was adding in the recent data. So new data gave us a new message, which changed our entire time series. For the tiger prawns, new data was useful, but in fact, there was a lot of how these guys using GLMs couldn't implement things like tensor splines, right? They were using standard isotropic splines. And it turns out that made a quantitative difference to the prediction of the model, right? So it was an interesting exercise to try to dissect, what was really driving these results. So here is another plot. What we're showing here is, again, catch differences, but this time we're doing sensitivity to the data. Essentially, this is the base is this guy here. Okay, and what you find is basically most of the time when we drop data, we get closer to the model that we had before. So the recent data was really influential, but the model structure is important as well. You might notice the crazy one, green line, that was when we used the data that we all would have liked to, which is basically only use data from when the fishery was operating. But remember that table about sample size? That reduced our data set by about 70%, right? So definitely some trade-offs here in terms of that. And as always, everything tiger prawns was more complicated. We also compared our results with the current assessment. Let's go to the next one. The other thing we started looking at was not just what's causing the change, but where is the change, right? This spot isn't all that easy to see. So this is semi-sulcatus. This is the tiger prawns, the endeavor prawns. You'll all remember that there were 7 areas. Does anyone remember where area 2 was? No, just north of Darwin in that Melville area. And remember, there was a big blob of green points above that island on the edge. And that's where we see all the changes in the endeavor catches were in that area. So it was purely a change in our predictions for that Melville area that then went through the entire catch series. For the tiger prawns, the main changes are in that area of the Gulf on the western side of the Gulf near that island that's boringly named Big. So again, a really interesting result that we were expecting that the changes would be sort of uniform across the space, but given the flexibility of a general additive model, it can actually be quite stable in some places and quite variable in others. So, okay, so the question is, who cares? Okay, so the stock assessment for the tiger prawns is unique in many ways. It involves a 3-species technical interactions model. So remember I said that the model creates effort, right? There is no effort data. There is nothing called effort by species. It's effort created by a model. So when we do the stock assessment, we actually run the 2 tiger prawn species in a single model with parameters shared between the species and interacting things. There's a biomass dynamics model for the endeavor prawns. The output does not go into Rick Method's harvest control rule, but actually goes into a completely separate bioeconomic optimization. So essentially the way this fishery management works is we estimate where we are today in terms of biomass, and then given predictions of next year's costs and prices, we find the effort level that will maximize profits. And so there's a whole lot of data that goes into this assessment that no other assessment in the world uses. And things like predicted fuel price, which by the way, really sucks. People can't predict next year's fuel price very well. And if you could, they wouldn't be working in academia, they'd be making real money. But prawn prices, all that kind of stuff. So the output of the model includes the stuff you're all familiar with, stock size relative to MSY, but also stock size relative to the stock size that maximizes profits. And the same thing with effort. The data, there's actually quite a lot of data that goes into the assessment. There's the catch and effort data we've talked about. There's survey data. There are actually 2 surveys a year. The fishery is very profitable. So the way it operates is it has a Le Mans star. You know what I'm talking about? Le Mans. Where's all these French people? Le Mans, the race.
No.
Sorry?
Doesn't sound French at all.
Isn't the rule that we get to say French the way? Anyway, what was I saying? So it's a Le Mans start. Literally all the boats are tied up in one harbour and there is a gun. And if I know Northern Australia, it probably is a gun. And they literally go out to sea and fish non-stop. until a mid-year closure where they basically stop fishing, go back to port, get fixed. And the way we've done our surveys is we do one survey before the fishery starts in April, and then when the fishermen are going back to have their boats sort of re-provisioned, we do a second survey. We've got tagging data to estimate growth rates, lots of length frequencies, really hard to get length frequencies of prawns on commercial boats because they want to freeze the catch. Economic data, and then data on prediction. So my own version of RFSS looks pretty pathetic, but you get a sense of the data. You can see most of our data starts here in the 2000s, except the tagging program was in the 1980s. So just a gentle reminder, here's a yield function. MSY is the maximum of the yield function, but we are maximum. So this is yield, but it would be the same as revenue, more or less. This is cost. The idea is we're going to find the level of fishing effort that maximizes the difference between the revenue curve and the cost function. And almost by definition, that is more conservative than MSY. So our effort levels are less than MSY. It is a multi-species fishery though, right? So that does mean you might overfish in a biological sense, one of the species to maximize catch, right? This is Australia, we catch fishermen.
Are there price differences between the species?
No, but they're productivity differences. Steepness is actually really low. You'll see that in a moment. So we run this model. I have no idea what time it is because I'm on the watch.
It's 1:30.
Oh, right. I'm supposed to do another meeting, but I'm almost done. So what did we find? So this is actually sort of almost the final straw. These are predictions of catch. You'll see the odd, I did say there are differences. The differences are in hundreds of tons out of thousands. So you're talking about a 10% change in catch. Enough to freak people out, but not catastrophically. And the predictions, these are predictions out for 15 years, I think, 30 years. And there are differences in terms of model predictions. That's biomass and catch. This is effort. This is effort relative to the target effort. You'll notice there are bigger differences when we look in terms of effort. This is an effort managed fishery, by the way. You can't calculate a TAC for effort. So TACs, we love them. Not so good if the animals you set in the TAC haven't been born yet, right? So you have to set a TAC before they spawn. Not so easy. So it's effort managed. And in fact, for those who are interested in fisheries management, it's actually head rope managed. By head rope. The length of your net is what you're managed in terms of. So when we change the quota, the fishermen have to cut parts of their net out to be able to reduce or increase effort. So you literally buy nets from people. It's a really nice way to manage effort because it's measurable. You can take a tape measure. So the final thing I want to show is this is our current model. And here's our best models. I want to have Alan have a breakdown because he's not going to believe that steepness value of 0.36. Unfortunately, that is what happens with prawns. And I just highlighted some of the changes between the old models, the new models. Essentially, things become more optimistic for the groove tiger prawns. We're predicting that they can fish harder, but the consequence of that being the law of conservation of matter is that we have reduced fishing effort on the other species. So we've actually changed, the way we do it is we manage where they're allowed to fish. So we basically close areas where the species we want to protect. So future work, this is all done using a GAM with a quasi-likelihood. There are no measures of uncertainty because of that. And so logical next steps are either get somebody to come up with a better piece of software, which doesn't seem likely because it turns out family equals quasi-binomial just doesn't work. We're probably going to bootstrap the whole thing eventually. Question is, what is the unit of bootstrapping in this data set? It sort of you'd like to bootstrap the whole study thing, but then you get a very unbalanced error. It's horrible. So if we ever get measures of precision, we want to then put those into the stock assessment to see what happens. Ultimately, we want to move to a mixed model, so something closer to a geostatistical model, but that's a long way away. And probably the most important thing from a management point of view is getting the managers to realize that data are important. So to collect those data virtually, we had to give our firstborn children up to get people on boats to collect data. What this is really saying is there really needs to be an ongoing monitoring of spatial mixed proportion data so we can update these models in the future. And preferably collect the data when there is a fishery, not when there's not a fishery. And with that, I'll stop. The plot on the right-hand side is pictures of people measuring prawns. And I really do need to acknowledge that without people spending hours and hours measuring the species and in fact sex of multiple prawns, this project wouldn't be done. And that is the project team. You may recognize some of these people. The person's smiling. Come on, who's got the best smile in fisheries? Who's that? Of course it's Eva Plagani. It's gotta be Eva. Who else has got a nice smile? The guy who's all grumpy is he's the biologist who's modeling. Anyway, I will stop there. Apparently, 10 minutes ago, I was supposed to be somewhere else. Questions?
You mentioned, I liked your future work slide. That was my question, was about uncertainty, and you mentioned that there are already some concerns, but not catastrophic concerns. Do you think when you bring in uncertainty, there may be some catastrophic changes, or as you put it, but larger changes?
Yeah, I mean, the question is, can we do the uncertainty correctly?
Right.
I mean, that's we can bootstrap, but if you bootstrap randomly with 35,000 points, you're going to get a really precise answer. And if you randomly throw out data sets, you're going to get unbalanced. Some of your bootstraps are going to have no data since 1990 or something. So it's hard to speculate exactly how big that effect is compared to, dare I say, predictions of variance in recruitment for an animal that lives 1 year, 1.5 years. So not sure where that's going to go.
Yeah, you said you wanted data collected while the fishery's happening.
Yeah.
Is that less important near the equator, where there's less of a seasonal effect?
There's still variation in temperature, and it does affect. What we're more sort of concerned about is extrapolation of behavior when the fishery is not operating because obviously it's a model, right? So you get data over here, it affects the output over there. So yeah, I mean, and there's just so much data that is out of season. But we can't throw it out because we'll run out of any predictive skill. Yeah.
Does the fishery seek a certain proportion, or they just go out and just go out?
The only time it comes into play is through eco certification. So they are required through their MSC certification to have all their stocks in the green zone, if you want to call it that, from a Kobe block point of view. So they don't care about until we say, well, brown tiger prawns are overexploited and you're going to lose your certification. Then they suddenly pay a lot more attention. But from a price point of view, the two species groups themselves, endeavors and tigers, have very different prices. Tiger prawns are worth a lot more than endeavors. But within the groups, the reason we've never really collected data is no one sort of cares. I actually had a paper rejected by an economist because I wrote a model that had the two species separately, and the economist said, well, they're worth the same. Why would you model them as species? Because they're product. That was when I stopped so many papers to economics journals. Yeah, oh, sorry, I think there was, yeah.
Explain a little bit more why you continue down the path of weight instead of converting the number if you have a length frequency that you can probably train with.
Because we have to convert weight, catch weight. Ultimately, our data, the thing that we've got in the logbook is catch weight. So our model prediction has to be in catch weight. And length, weight, I mean, in principle, we could work in numbers, but then you need to know the length frequency, and it just gets complicated. We got survey length frequency,
Okay.
but not some of those. We lose almost all of the early data. I mean, the early data was literally, you get people taking filing cabinets and looking up old data sheets, and yeah, data tends to disappear. Yeah.
Doing meteor management
decision when they will go back to their recreation.
Mhm.
At the time, they consider how much to fish, and that's how it works.
No, they can fish for as long as they like.
Okay.
But the only thing they're restricted in terms of the season, that's one of our management controls. Is we control mainly the length of the head rope, but we can also stop the fishery at different times. So I didn't get into it, but generally speaking, the early part of the season tends to be the brown tiger prawns, and later groups. So if we want to restrict browns, we can close areas. All the nursery grounds are closed, for example, but we can also stop the fishery operating in the first season, so that then that affects their target.
And if you know you have the seasonal pattern of early in the season, you have I don't see any seasonal pattern anymore. Like, why is your only day thing is a day since the fishery
No, there was a day during the year as well.
Oh, okay.
I probably forgot. That was one of the
I
covariates.
feel like you only have days since.
No, there's the temporal signal is a spline in terms of cyclic spline within the year, and then an annual spline on top of that. And there is, it's one of the within season spline is, not just supported by, fancy modeling, but actually by experiments where you actually watch, you change the temperature and the behavior of the species changes.
And why a day since, why is your time measurement is day since the fishery started, whenever,
Mhm.
not a year?
Because we're making predictions by day. And more importantly, December the 31st is not different from January the 1st, right? So we wanted everything continuous.
Do you read by week, like do you read the days?
Well, yeah, it's a model. But yeah, I mean, it does mean we have to pin the data down literally today, so.
I think Jim Hasty had a similar question online where he asked, did you consider using a longer period than a day for the modeling, given the nature of the data?
Yeah, it wouldn't have mattered really. It'll just be what your prediction is. You're not going to get increased precision by doing that, because you're still going to have a spline. Instead of reading off day, you'll be reading off week. It'll run fast. But, as I've said before, computers have no rights except the right to work. So,
Okay, I think already 10 minutes past your meeting time. Oh, I think Noble has another one, but Noble, can you maybe follow up on email? Relate to a meeting, but I just want to say thank you everyone for coming to Quantum today, and we hope to see you next week. We're going to hear from Ray Hilborn on reducing the carbon footprint of fishing, the roles of fisheries management. So thank you, everybody.
thanks, everybody.
Thanks, Andre.
And next year, it might be Kristen giving this call, if she's got her job in Hobart.
One trout for water.
I know.